We'll demystify CRISPR-Cas9 gene editing, how bacterial immune systems became precise molecular scissors, how guide RNAs target DNA, and why this technology is transforming medicine, agriculture, and bioethics today worldwide. Foundations: DNA, genomes, and the rise of CRISPR. All life stores information in DNA. The human genome contains about 3 billion base pairs and roughly 20,000 protein-coding genes. For decades, researchers used restriction enzymes, zinc finger nucleases, and talins to cut DNA, but these methods were complex and costly. In 1987, researchers observed clustered repeats in bacterial genomes. By the 2000s, these became known as CRISPR, clustered, regularly interspaced, short palindromic repeats, linked to Cas, CRISPR-associated proteins. In 2012, Jennifer Doudna and Emmanuel Charpentier showed that an engineered guide RNA can program Cas9 to cut specific DNA sequences, creating a fast, programmable editing tool that revolutionized genetics. Mechanism: how CRISPR-Cas9 finds and edits DNA. CRISPR-Cas9 editing uses two main parts: a guide RNA, gRNA, of about 20 nucleotides that base pairs with a target DNA sequence, and the Cas9 nuclease that cleaves DNA adjacent to a short PAM sequence. For Streptococcus pyogenes Cas9, the PAM is NGG. When Cas9 creates a double-strand break, cells repair it by non-homologous end joining, NHEJ, often producing small insertions or deletions, indels, or by homology-directed repair, HDR, if a donor template is supplied. Typical on-target editing efficiencies vary widely, from approximately 10% to greater than 70% in different cell types, while HDR rates are generally lower, often below 10% in primary cells. Applications and concrete examples: CRISPR is already in clinical and commercial use. Ex vivo therapies modify patient cells outside the body. For example, CRISPR-modified hematopoietic stem cells targeting the BCL11A enhancer have produced functional cures in sickle cell and beta thalassemia trials. In vivo approaches delivered by lipid nanoparticles, or AAV, are advancing. NTLA 2001, an LNP-delivered CRISPR therapy, showed up to approximately 90% reduction in transthyretin protein in early human data. Agriculture uses include non-browning apples and disease-resistant crops. Diagnostics harness Cas12, Cas13, detector, Sherlock, for rapid viral detection, including SARS-CoV-2 assays. Advanced methods, risks, and ethical implications. Newer tools include base editors, change single bases without double-strand breaks, and prime editors, programmable small insertions, deletions, and all base-to-base conversions. Prime editing was reported in 2019 and expands precision. Key risks are off-target edits and mosaicism, immune responses to Cas proteins, and delivery limits to specific tissues. Ethical concerns center on germline modification, crystallized by the 2018 announcement of edited babies, and environmental risks from gene drives. Governance is evolving. Research moratoria, regulatory review by agencies like FDA/EMA, and international discussion aim to balance innovation with safety and equity. CRISPR transformed a bacterial defense into a precise toolkit for rewriting DNA. Its mechanism, guide RNA targeting and Cas-mediated cutting, enables diverse applications from cures for genetic disease to agricultural improvements. Responsible deployment requires attention to delivery, specificity, safety, and ethical governance to realize benefits widely.